In this lecture, we're going to talk about transporting molecules across the membrane, like through diffusion, osmosis, facilitated diffusion, etc. And then we're going to focus more on osmosis, the movement of water, and how solutes can affect the movement of water in and out of the cell. So we're going to talk about hypertonic, hypotonic, and isotonic solutions. Not all molecules are able to cross the membrane. So we say that the membrane is semi-permeable or selectively permeable. Now, whether these substances or these molecules are able to cross is due to the properties of these molecules and also the properties of the molecules that make up the plasma membrane. Remember we said that the phospholipids have heads that are polar, they're hydrophilic, they'll interact with other polar molecules like water, and then we have those nonpolar tails, they're hydrophobic. The tails of the phospholipids are going to interact with other nonpolar molecules or hydrophobic molecules and are not going to interact with polar molecules. So it's really the properties of these molecules that make up the plasma membrane that are responsible for this semi-permeable or selectively permeable nature of the plasma membrane. Now, it's important to have an idea of some molecules that need to get into the cell and examples of molecules that need to come out of the cell. And that's what this diagram is showing you. So the cell needs to take in lipids for energy, amino acids to build proteins, sugars for energy for cellular respiration. Some cells need to get rid of molecules like CO2. Animal cells need to get rid of that excess CO2. Ammonia is considered a waste. Ammonia is produced by the breakdown of amino acids. And ammonia is a major component of urine. It gets converted into urea and then it gets excreted as urine. So it's a waste and it's going to be toxic to cells, so cells have to remove that waste. So molecules are constantly moving into the cell and out of the cell. Now, these molecules or these substances or these ions can either move directly through the phospholipid bilayer or they're moving through some kind of transport protein, or they're being moved in and out of the cell through vesicle transport. And we'll talk more about these methods here in a little bit. Now, in this diagram, I've included different substances that can move through the membrane and the properties associated with these different substances. And it's important that you're able to tell me for these different molecules and substances how they're going to cross the membrane and why they have to use that particular method. Now, molecules that are crossing through the phospholipid bilayer, sometimes we say that these are the molecules that can easily move through the membrane. And what we mean by that is that they're simply just moving through the phospholipids either in or out of the cell. In pre-AP, we learned that the substances that can easily move or move through the phospholipids in and out of the cell are the ones that are small, uncharged, and like the plasma membrane. And what we mean by this last one is that the plasma membrane primarily is made of these phospholipids or fats. And so fats can easily cross the membrane. So they're molecules that are like the membrane molecules. And in pre-AP, we compared this to getting into a club. Okay, so imagine if you were selecting the people to come into the club. And it's your job to remove these people if they're causing a disturbance. So think of the people that you would select to come into the club, the ones that can easily move into it. They would be small because if you have to throw them back out, it would be nice if they were smaller than you. They need to be uncharged. If you see someone and they're all charged up, it can mean that they're a little unstable. They might be a little dangerous. They might cause issues when they get into the club. And you're more likely to be trustworthy or allow people in that look like you. If they're like you, then you might be able to relate to them easier or reason with them. So if you have problems trying to remember the molecules that can easily cross, then try to remember that example. Small, uncharged, and like can easily cross the membrane. So let's add some answers to these blanks in this box. So molecules like lipids can easily cross. These are nonpolar molecules. And they're going to be hydrophobic. Nonpolar hydrophobic molecules are lipid soluble. that means that they're going to be able to cross through that phospholipid bilayer. Molecules like gases, like oxygen and CO2, are able to cross that phospholipid bilayer because they're very small molecules and they're uncharged. So these nonpolar molecules that, like lipids, are just going to be able to cross through the phospholipid bilayer because they can interact with those nonpolar tails. And in this case, for our cells, we would be bringing oxygen in, and we would be getting rid of CO2 because it's a waste of respiration. Now, molecules that aren't lipid soluble are going to have to cross the membrane with some assistance. They're going to need a specific transport protein. And we said there's different types of transport proteins. We can call them channels, carriers, or pumps. So molecules like glucose that are polar are going to have to go through a transport protein. These molecules are hydrophilic. They will interact with water molecules, but they will not interact with these nonpolar tails of the phospholipid. And ions are going to need a transport protein as well because they have a charge, and we indicate that with the positive and negative charges behind their symbols there. And because ions are charged, they're going to interact with polar molecules, but again, they're not going to interact with these nonpolar tails of the phospholipids. So these molecules that I indicated here in ions are going to have to move across the membrane through some kind of transport protein. And I want to point out again that only specific transport proteins can transport certain ions. So protons can only go through a certain type of transport protein. Chloride ions would have to go through a different type of transport protein. Water molecules go through a transport protein that we call aquaporins. So these transport proteins are really specific to the certain substances that are moving across the membrane. Now, before I go on to the next page, I just want to talk about this box. And this is a learning objective that is from the course description. So this kind of reminds me, and it indicates to you what you are expected to know for that AP test. So you should be able to explain how the properties of the molecules that make up the cell membranes will affect the different molecules that can cross the membrane. Now, ultimately, I want to be able to give you an example of a molecule, maybe indicate some properties about that molecule, and you should be able to predict what type of transport is going to be used to move that molecule across the membrane. And you have 6 to choose from. There's diffusion, osmosis, and facilitated diffusion. And then we have active transport, endocytosis, and exocytosis. So these are the 6 main types of transports that we're going to cover. Now, I've written them like this because they can either be categorized as passive or active types of transports. And you need to remember that diffusion, osmosis, and facilitated diffusion are considered passive transports. Whereas active transport, endocytosis, and exocytosis are considered active processes. Now, anytime passive transport is occurring, that means that the cell is not using any energy. No ATP is used by the cell if a molecule is being moved via diffusion, osmosis, or facilitated diffusion. Also, substances, molecules, ions, if they're being moved passively by one of these 3 different methods, they're being moved down, or we say with, the concentration gradient. Or another way to say this is molecules are moving from high concentrations to where they're in low concentrations. So I like to envision this diagram. We're thinking of passive transport. We have areas of high concentration, low concentration. So imagine a hill, a high area and a low area. And molecules are going to be moving with the gradient or down the gradient from high concentrations to low concentrations. Now, for the transports that we listed on this side, these three types of transports require ATP. So the cell has to use ATP to move molecules by either active transport, endocytosis, or exocytosis. And here I want to point out that only for active transport can we say that molecules are being moved opposite. Instead of down the concentration gradient, molecules are being moved up the concentration gradient, or in this case, active transports moving molecules from low concentrations to high concentrations. That isn't exactly the case with endocytosis and exocytosis. So let's start with passive transport, and we said that, again, molecules and ions are being moved in and out of the cell, and it does not cost the cell any energy. These molecules or ions are being moved with the gradient or down the concentration gradient, which means that they're being moved from areas where they're in high concentration to areas where they're in low concentration. Now, you've probably heard that molecules, they're going to have a net movement from high to low until equilibrium is reached. And this is what we mean by equilibrium has been reached. So you can see that it looks like there's an equal amount of these substances on both sides of the membrane. But I want to point out a few misconceptions. Students think, like in the center diagram right here, that these molecules are only going to be moving in this direction. And it's more a net movement. We're going to have more molecules moving in this direction than in the reverse direction. But if this molecule collides with the membrane, it could go across. But again, there's more molecules moving in this direction. So we like to say that there's going to be a net movement of molecules until equilibrium is reached. So again, whenever this situation is reached, equilibrium is reached, we're not going to have a net movement of molecules in either direction. We're going to have more of an equal movement of molecules. Some molecules are going to cross this way, and some molecules are going to cross this way. So again, there's no net movement going on whenever we see equilibrium has been reached. So here we have a diagram that shows you these three types of passive transport that we just put on that first concept map. We have diffusion, osmosis, and facilitated diffusion. All three of these are considered passive transport, so that's why you see this in the diagram. And you can see these molecules are all moving from areas of high concentration to areas of low concentration. So here's an area of high concentration versus low concentration, so these molecules are moving in this direction. Same with here, high concentration to low concentration, high concentration to low concentration. Now, from what we just discussed, you should be able to give me examples of molecules that are moving by this method, diffusion. And diffusion is happening whenever these molecules are moving right through the phospholipids. And we said that molecules that are able to do this are molecules that are lipids or nonpolar molecules. These molecules are often small and they're uncharged. So this dot could be a lipid like testosterone, which was a lipid hormone, or this dot could be oxygen or CO2. Each of those molecules has the ability to interact with these nonpolar tails and cross through the membrane. Now, if osmosis is occurring, then that means that water is moving through the membrane or across the membrane. So those squares represent water molecules. And remember, this transport protein is called an aquaporin. If facilitated diffusion is occurring, then that means we have molecules or ions moving across the membrane through a transport protein. And these substances that are moving are charged. So we can have ions like potassium, chlorine, sodium, or these might be polar molecules. They cannot interact with these nonpolar tails, and so they have to cross through a specific transport protein. Okay, let's summarize what we just talked about on that previous diagram. So diffusion is the movement of small, uncharged, lipid-soluble substances or nonpolar substances across that phospholipid bilayer from areas of high concentration to where those molecules are in areas of low concentration. Osmosis is the movement of water across the membrane from high concentrations of water to low concentrations of water, and that channel protein that allows for that is called an aquaporin. And the reason why water molecules, like you can see here, are able to move through that aquaporin is because it has charged regions. And you can see those charges indicated right here on that diagram. So there's charged areas of the aquaporin that's going to interact with the charges on those polar water molecules. And then finally, facilitated diffusion aids in the movement of ions and mid-sized polar molecules from areas of high concentration to where they're in low concentration. And I said that these transport proteins are specific to substances that they're transporting. They have to have the right shape and the right properties to bind to these molecules that can move across the membrane. I included this diagram. So again, you could see what simple diffusion looks like. And then these diagrams down here are showing you facilitated diffusion. And again, you see that molecules are moved from high concentrations to low concentrations. But I wanted to include this just so that you can see the difference between how channel proteins work versus these carrier proteins. And this part of the diagram down here shows you how a carrier protein works. Channel proteins, they span the membrane, and they're more of an open channel that allows for the movement of specific molecules to cross, whereas these carrier proteins are going to go through a shape change. So the three pictures that you see here are not three separate carrier proteins or transport proteins. It's just showing you how this one is going to change once this molecule binds to it. So once bound, there's a shape change. And delta means change. So there's a shape change, and that allows for this molecule to cross the membrane when that shape change occurs. And I also wanted to take the time here to show you that the amount of transport proteins isn't always constant in a cell's membrane. And you can see that here. So we've talked about how your body has to regulate the amount of glucose. And another molecule that's involved with that is insulin. And insulin targets your liver cells and your muscle cells. I'm going to put liver cell. So here's the membrane of that liver cell. When insulin binds to a receptor protein, then it's signaling that cell to take up extra glucose. So let's put some glucose molecules on there. And insulin is triggering the uptake of glucose because there's vesicles. And these vesicles have transport proteins embedded in them. And specifically, those are those GLUT transporters that we talked about. And those are just transport proteins that can allow glucose to cross the membrane. So once insulin triggers that receptor, it stimulates these vesicles to move to the membrane, and we now have these GLUT transporters embedded. And so then glucose can move into the cell through those GLUT transporters. Now let's move into those three types of active processes: active transport, endocytosis, and exocytosis. So first, with active transport, molecules are going to be moved from an area of low concentration to an area of high concentration. And they're being moved through a transport protein similar to facilitated diffusion, just backwards. In facilitated diffusion, these molecules and ions were being moved from high to low. In active transport, they're being moved from low to high, like you can see here. So here's a lower concentration compared to the other side of the membrane where there's a higher concentration of those molecules. And you can see that it's going to cost the cell some ATP, or some energy. Now, sometimes these proteins are referred to as pumps. Think about what a pump in the basement does. It moves water from a low area in your basement to a higher area. And that's what's going on in this situation, but we're moving molecules from low concentrations to high concentration. So that's why that protein is sometimes called a pump. So if molecules are being moved from low concentrations to high concentrations, then we say that they're being moved up or against the gradient. Remember that diagram I showed you? High, low, and we're moving up or against the gradient. And this is going to require an ATP molecule every time these molecules are moved across the membrane. So now I want to take a second to introduce to you the sodium-potassium pump, because the sodium-potassium pump is doing active transport. It's moving sodium and potassium ions against their concentration gradient. And this is occurring in your nerve cells. And if this didn't occur, then your nerve cells wouldn't be able to transmit a message. So in this diagram at the top, it's showing you what a sodium-potassium pump does. And it's showing you 1, 2, 3, 4, 5 different proteins in the membrane, but it's actually just one. And they're just showing you what happens over time as these ions are binding to this transport protein. So we can see that sodium here binds to this pump, and eventually those sodium ions are going to be pumped out of the nerve cell. At the same time, the active site is being changed in the sodium-potassium pump, allowing these potassium ions to bind, and they're being brought into the cell. Now, when we get to the nervous system, when I talk more about the sodium-potassium pump, it helps sometimes if students remember that sodium, you probably heard of, is bad for you in excess. And that's true, but you do need sodium in your diet so that the sodium-potassium pump can set up this concentration gradient. But you might think people tell you to restrict your sodium, keep your sodium out of your body. So that usually helps students remember that sodium is being moved out, so keep it out. And you probably heard that if you eat bananas, you're getting potassium. Potassium's good for you. So potassium is always being brought into the nerve cells. And again, that's just a trick to help you remember the direction in which sodium and potassium is going to be moved across the membrane of these neurons. So you can see here that we're setting up this concentration gradient with a really high concentration of sodium on the outside, yet the cell continues to move sodium out from low concentrations of sodium to high concentrations of sodium. And we're setting up an area of high concentration of these potassium ions inside the cell. And the cell continues to move potassium in from areas where it's in low concentration to areas where it's in high concentration. If we are referencing vesicular transport, then we're probably talking about endocytosis and exocytosis. Now, these are considered active processes because they cost the cell energy. And vesicular transport is used for substances that are just too large to fit through a carrier protein or a channel protein. Now, I want to point out that it's not costing the cell energy because substances are being moved from low concentrations to high concentrations. Substances might be moving from high to low concentrations or low to high, but the energy need comes from the movement of the cytoskeleton to move the membrane so that these vesicles can form. So that pseudopods, remember we talked about pseudopods are like extensions where the cell membrane can wrap around substances. We mentioned pseudopods when we were talking about protists, and we said that amoebas, so I'm going to put protist, not all of them, but one example is an amoeba, forms these pseudopods and wraps around these food particles, forming a vesicle to bring those food particles into the cell for digestion. So when the cell membrane is being moved and manipulated to bring in these substances, we say endocytosis is occurring. So endo means in. So the movement of macromolecules into the cell by enclosing them in a portion of the membrane. Remember, macro means big, so we're talking about big molecules moving into the cell, but cells just aren't bringing in macromolecules. Our white blood cells are a type of cell that's engulfing an entire cell, like a bacteria cell. So it's just not engulfing a big molecule. It's bringing an entire bacteria cell into the cytoplasm so it can be digested. So our white blood cells are using endocytosis. And I wanted to show you 3 different terms that are related to endocytosis. We talked about pinocytosis, whenever large dissolved solutes are being brought into the cell. Phagocytosis is whenever there's large non-soluble substances being brought into the cell. And then we haven't mentioned yet receptor-mediated endocytosis. So in this instance, there's a cell that has a receptor protein on its surface, and whenever these solutes bind to these receptor proteins, like you can see has happened here, then that triggers the vesicles to form. So these molecules can be brought inside of the cell. And you can probably guess that when we're talking about exocytosis, we're talking about the movement of substances, large substances, macromolecules, out of the cell. An example of a molecule that would be released through exocytosis would be a protein hormone. Proteins are often big molecules, and they themselves wouldn't be able to fit through a transport protein. I included this diagram here to show you that the phospholipids that make up this vesicle are just going to become phospholipids that make up the plasma membrane when that vesicle fuses with the membrane. And then I've included these diagrams here just to give you another look at vesicular transport. So you should be able to determine that we have endocytosis going on here. And here's an example of exocytosis. And again, here we have endo, and here we have exo. After going through that information, I should be able to remove all the text from these boxes, and you should be able to fill in this information. So I've just summarized the type of molecules that are moving by diffusion, osmosis, and facilitated diffusion, and how these substances are crossing the membrane. Either they're going through the lipid bilayer, or maybe they're going through a transport protein. But I wanted to point out here that facilitated diffusion and active transport are pretty much opposites of themselves. So if you're memorizing the types of molecules that are moving by facilitated diffusion, then you have them memorized for the molecules that are moving by active transport. It's just whenever these molecules are moving from high to low, then we call it facilitated diffusion. When these molecules are being moved from low to high, then we say that active transport is occurring. And I see that I've made a mistake here because protein hormones often aren't taken in by endocytosis. But we talked about like bacteria are taken in by white blood cells, and food particles are taken in by protists. Protein hormones are being excreted, so they're moving through the membrane, usually by exocytosis, as they're leaving the cell that made them. And again, we have big molecules being moved in and big molecules being moved out. So that was a quick overview of those 6 types of transports: diffusion, osmosis, facilitated diffusion, active transport, endocytosis, and exocytosis. And those 6 can either be categorized as passive or active. Now we're going to focus more on osmosis. And the reason why the next pages are dedicated just to osmosis is because organisms have to very carefully regulate their water and their solute concentrations, because that can affect whether water is moving into their cells or out of the cells, and that can be the difference between life or death. If too much water moves into cells, then animal cells can rupture. If water leaves the cell, then the cells are going to die. Ultimately, I should be able to give you a description of 2 different solutions, and you should be able to predict which way water is going to move. So there's 4 different rules that you might have to apply to different diagrams. So rule number one is that water and all molecules always move from areas of where they're in high concentrations to areas where they're in low concentrations. So let's reference this diagram first. You can see that there's more water molecules on this side of the membrane compared to this side, so you should be able to predict that water is going to move in this direction, or there's going to be a net movement of water from left to right in this diagram. Sometimes you're just given percents, so you need to understand that 90% is a higher concentration of water than 50%. So here's a situation that the water molecules are going to move in this direction. They're going to move from areas of high concentration of water to areas of low concentration of water. Sometimes you're not given water percents, and you can see that in diagrams down here. There's no water molecules pictured, and it doesn't give you any indication of water concentration or solute concentration. In these diagrams, they're giving you different types of solutions. So you're going to have to memorize and apply rule number 2. You need to remember that water moves from a hypotonic solution to a hypertonic solution. Now, the easiest way to remember this is what I taught you in pre-AP. Think of going to a party this weekend. Do you want to go to a party where everyone's really hyper or where everyone is really bored? Of course, you want to go to the hyper party. So remember that. Water wants to go to the hypertonic solution. Now, let's talk a little bit more about what a solution is. In a solution, we have solutes and then we have the substance that's doing the dissolving, the solvent. And in many solutions, it's water. So the dissolved molecules are called the solutes, and water is considered the solvent. And in the last unit, we talked about that it's noted as the universal solvent because it can dissolve so many different molecules. Now, if we have a hypertonic solution, then that simply means that there's a higher concentration of solutes or dissolved substances in that solution compared to another. The solution that has a lower concentration of solutes is called the hypotonic solution. So hypotonic solutions are solutions that have a lower concentration of solutes. If you have two solutions that have the same concentration of solutes, then they're considered isotonic solution. Iso means the same, like think of an isosceles triangle, a triangle that has two equal sides. So two solutions that have the same concentration of solutes are termed as isotonic solutions. So let's apply rule number 2 if you're given a situation like this. So it says that there's a solution in the beaker and that solution is hypertonic compared to the solution that's in this dialysis bag. And hopefully by this point in class, you guys have had a situation to set up your dialysis bags in a solution. So you have an idea of what this diagram is about. So the question is, if water is the only molecule that can move through the dialysis tubing, in what direction is there going to be a net flow of water? So apply that rule. Water moves to the hyper party. So the outside is hypertonic, the inside's hypotonic. Water is going to leave the dialysis tubing. Let's apply that to these diagrams here. So here we have cells. And it's telling you that the cells are placed in these solutions. So it's saying that the outside of the cell is hypertonic. By default, that means that the inside of the cell is hypotonic. So in this situation, water is going to move to the hypertonic. Remember, go to the hyper party. So water is going to move out, or there's going to be a net flow of water out of the cell. If the outside solution is hypotonic, then the inside's hypertonic. Water is going to flow into this cell. And there's no net movement of water occurring whenever the outside solution is isotonic compared to the cell. So for the water that's going to move in, there's going to be a relatively equal amount of water that's moving out of the cell. Now, one thing that I want to note is that if we term a solution as being a hypertonic solution, that label isn't stuck to that solution forever. It's a comparative term. So if I say that I'm short, I'm going to be short compared to most of my students. But I'm not always short, because if you can compare me to a preschooler, then I'm going to be tall. So if you're determining whether a solution is hypertonic or hypotonic, you have to have another solution to compare it to. So here we have a solution that has no sugar in it. So it's pure water. So it's 0 molarity sugar solution. Compared it to a 1 molar sugar solution. So in this situation, this one would be hypo. This one would be the hypertonic solution. Let's take that exact same 1 molar sugar solution, and here it is. Let's compare it to a 3 molar sugar solution. So now that solution that we just said hypertonic is now hypotonic, whereas this solution is considered the hypertonic one. Now, for these diagrams that you see here, the first two rules are not going to be usable in these situations because these diagrams are just showing you solutes. So rule number 3 states that if you're just given the amount of solutes in a solution or you're just shown the solutes, then you need to remember that water moves to where there are more solutes. So think of this as solutes suck. Wherever there's more solutes, that's where water is going to go. Now, I set up a demo in class sometimes if I have a lot of extra Halloween candy, because I'm usually talking about these concepts around that time. And so I put this big box of sugar candy on one side of the room, and I tell my students, you guys are all water molecules. Where do you want to go? And every student wants to go dig through that box of candy. So think of that candy as being sugars, salts, solutes, and solutes are going to suck all the people to it, or they're going to suck all the water molecules to it. Another way to remember this concept is think of if you eat some really salty potato chips. In about 15 minutes, you're going to be very thirsty. You took in all those salts, all those solutes, and pretty soon you're going to be sucking down a bunch of water to try to maintain homeostasis. So solutes suck water. In this first diagram, you see that there's more solutes over here, and that's what these words down here are indicating. There's more solutes on this side of this U-tube. So if this is a semi-permeable membrane and that water can cross, then water is going to move to this side of the U-tube. And the water level on that side is actually going to go up, while this side is going to drop. In this diagram, you can see that we have two solutions in some kind of container, and those solutions are separated by a semi-permeable membrane. And you can see that there's more sucrose molecules dissolved in the solution on side B. So let's call this side A and side B. And so you can apply the rule that solutes suck. There's more solutes on side B compared to A. So water is going to move from side A to side B. And so there's going to be a net movement of water molecules from A to B. Now, I do want to point out that water molecules still can move from B to A, but you're just going to have a greater flow of water molecules moving in this direction compared to this direction. So that's why you still see these smaller arrows indicating that water can move in both directions, and then you have this bigger arrow that's just indicating the direction of the net flow of water. Let's do one more example. So I'm going to put a beaker here, and I'm going to add a solution to it. And the concentration of this solution is going to be 3 molar. And again, this M is just a measurement of concentration. It means moles per liter. So if you see that M, we're just giving you some kind of indication of solute concentration. So I'm going to put a 3 molar solution in the beaker, and then I'm going to put a solution in some dialysis tubing and tie it off. And let's say the concentration of the solution in the dialysis tubing is 1 molar. So you need to understand this is solute concentration we're dealing with, and solutes suck. So wherever there is a higher concentration of solutes, that's the direction that water molecules are going to move. So in this situation, water molecules would move out into the solution in the beaker. And if we took the mass of this dialysis tubing before and after, we would see that the mass of it decreases because the water is moving out of the tubing. Okay, moving on to the next page, you see rule number 4. And rule number 4 is new. It wasn't taught in pre-AP. And rule number 4 has to do with water potential. And we're going to have an entire lecture that's dedicated to your understanding of water potential. So there's more coming about it, but I just wanted to introduce it here. So the rule that you need to remember is that water moves from a solution with a higher water potential to a solution that has a lower water potential. And that should be easy to remember because we keep indicating that molecules move from high to low, and we've said high concentrations to low concentrations. Just apply that high to low to water potential. So water is always going to move from a solution with higher water potential to a solution with lower water potential. Now, I have a diagram right here, and this is called a U-tube, and it's giving you the water potential measurements for two different solutions. So I have a solution on the side, we can call it side A, and here is side B, and in the middle there's a semi-permeable membrane that water can cross, but the solutes can't. Now, the symbol for water potential is this psi sign with a W behind it. So here is the measurement of water potential for side A. So I'm going to put a little W there. That's water potential. And then the water potential for side B is down here. And later on, I'm going to tell you what these two numbers mean. So we have a water potential. So let's write water potential on this side is 0. And on side A, we just have pure water, and something to have memorized eventually is that the water potential of pure water that contains no solutes is always going to be at 0. And on side B, obviously we have solutes because you can see them drawn in here. And it's been determined that the water potential for this side is -1. And water potential can be any value. It can be a positive value. It can be a negative value. In this situation, it's a -1. But I want to make sure that you understand that not all solutions that have solutes in it are always going to have a water potential of -1. So this can change. But for this situation, we have a water potential of 0 and a water potential on this side of -1. And you just have to apply the rules that water moves from high water potential to areas of lower water potential. So 0 is higher than -1. So water is going to move in this direction. So now I want to work through some different examples where you can choose which of these rules to apply to determine the direction that water is going to move. So in the first example, this diagram right here, number 1 says the dialysis bag contains a 0.2 molar starch solution. Assume starch cannot cross the membrane, indicate the net direction of water flow. This is really similar to the beaker that we just drew on the previous page. So this is a concentration of solute. So you have 0.2 versus 0.8. And sometimes decimals throw off students. So for me, to make this easy, I think of this as 20 cents, right? 0.20 would be 20 cents. And this is 80 cents. And what do you want more of? You want 80 cents because it's a higher value. So this is indicating there is more solutes on the outside compared to the dialysis bag. And water always moves to where there's a higher concentration of solutes or into a hypertonic solution. So water is going to move out in this case. For question number 2, it says for diagrams A and B, indicate with an arrow the net flow of water. So let's just start with A. We have two solutions that are isotonic to one another, so there's not going to be a net flow of water in either direction. So we can't add an arrow to A. Taking a look at B, though, they give you information like this side's hypotonic, this was hypertonic, so you can just apply the rule that water always moves from the hypotonic solution to the hypertonic solution. Remember, you go to the hyper party, water's going to go to the hypertonic solution. So we know water's going to flow in this direction. You could have also applied the rule that solutes suck, and you can see from the key here, the big circles are the solutes, and there is a greater concentration of solutes on this side compared to this first side. So again, water goes to where there's more solutes, so it's moving in that direction. And then you could even look at it from a water standpoint, and we have a higher concentration of water over here. If you count the water molecules, there's 1, 2, 3, 4, 5, 6, 7, 8, 9 out of 10. So this is 90% water on this side. It's about, let's call it, it's about 60% water on this side. And so water moves from areas of high concentration of water to areas of low concentration of water. So water would be moving in that direction. So it doesn't matter which rule you apply, water is always going to be moving in the same direction, regardless of the rule. It's just sometimes you're not given water percent, so you can't use the first rule. Or you're not given the solution type, hypertonic or hypotonic, so you can't use that rule. Sometimes you're not given solute amount, so you can't use that rule, and you're going to have to apply other rules. So let's take a look at this last example here. And this one's tricky because it factors in a few different concepts. Some concepts from biochemistry when we talked about monosaccharides and disaccharides. So it looks like we have an artificial cell. So the circle is representing a cell that's in a solution. And this has given us an indication of the amount of different solutes that are dissolved in the outside solution and then in the solution inside the cell. So it says, predict the solute amounts after equilibrium has been reached if only monosaccharides can cross the membrane. So before you can do anything, you have to understand what a monosaccharide is. So sucrose is a disaccharide. Remember, it's a glucose plus a fructose molecule joined together. And since it's a disaccharide, it's saying here that it can't cross. Only monosaccharides can. So we're going to lock in these amounts for sucrose because they're not going to change because sucrose can't cross the membrane. And we really didn't discuss maltose much in the biochemistry unit, but maltose is a disaccharide. It's two glucoses joined together. So we're going to lock in the maltose amount because it can't cross the membrane. So we're only going to allow the monosaccharides to move. And remember, all molecules, if they're allowed to move, are going to move until they reach equilibrium. So we need to determine what would be the amounts of glucose outside and inside the cell once it's reached equilibrium. To do that, let's figure out the total amount of glucose that we're dealing with. So we're simply going to add these two amounts together. So 0.02 plus 0.005, and that equals 0.025. So we have that to split between the two different solutions. So take that number and divide it by 2, and that gives us 0.125 would be in the inside, and 0.125, and I'm guessing this is probably dealing with grams, so 0.125 grams of glucose would be on the outside. Fructose is a monosaccharide, so it's going to move until it reaches equilibrium in both solutions. And if this is our total amount of fructose, then we just simply have to divide that by 2. So 0.01 divided by 2 gives us 0.005. And so I'm going to put 0.005 inside, and that's for fructose. So if you have room and you want to put an F there for fructose, that's giving you an indication of the amount of fructose inside the cell. Now, this fourth question says, indicate with an arrow the net flow of water. So to figure out which way water is going to go, we need to figure out where there's more solutes, because that's going to be the hypertonic solution, and that's the direction in which water is going to flow. So we just need to add up our values of our solutes. So we're going to add up 0.2, 0.125, 0.005, 0.01, just to figure our total for solutes in the outside solution. And that value is 0.331. If we add up the solutes inside the cell, that gives us about 0.63. So I'm going to write that up here, 0.63. And when we compare those two values, obviously there's more solutes inside the cell, so we're going to have a net flow of water into the cell. On this page, we're going to talk about some concepts related to the dialysis tubing lab that you assembled. Remember, you were given starch and glucose, and you were asked to make two different solutions, one to go into dialysis tubing and then one to go into a beaker in which we're going to place that dialysis bag. Some concepts this lab addresses is how molecule size affects its ability to move through a membrane. Like dialysis tubing, it's a membrane. So if you think of starch, starch was a polysaccharide. It's made of thousands of glucose subunits. So starch is a really big molecule compared to glucose. Glucose is just a monosaccharide. There's small pores in the dialysis tubing, And so just think about which molecule is going to be able to cross. And we're going to be able to identify this by using the glucose testing strips and using iodine to see if starch is in the solution, because when we add iodine, it's going to make the solution turn purple if there's starch in that solution. And then we're also examining how solute concentration can affect the movement of water. So we had a beaker, and then we had the dialysis bag in a solution. And you were asked to take the mass of the bag. So next class, when we're taking the final mass of the bag, if water has moved out, then obviously the mass of the bag is going to decrease. And water would move out if you created a solution where there was a higher concentration of solutes on the outside compared to the inside. So just depending on the solutions that you created and where you placed them, either into the bag or into the beaker, some of you are going to get the mass of your bag increasing as water moves into the bag, and some of you are going to get the mass of the bag decreasing if water is pulled out. Now we're going to talk about calculating percent change in mass. You're going to be asked to do this on the worksheet that goes with the dialysis lab, and then you're also going to have to do this in a few other labs that we're going to finish up the unit with. On your formula sheet, there is no formula for calculating percent change in anything. You're going to have to think about it and reason your way through some of these calculations. If you just want to memorize this formula, you're more than welcome to, but I think a lot of you are just going to be able to reason how you should go about calculating percent change in mass. So percent change in mass is final minus initial, and then you divide by initial. You're going to take that number and you're going to multiply it by 100. And that's going to give you a percent. I'm going to plug in numbers into this equation, get an answer, and then I'm just going to walk through reasoning how to solve for this problem if you can't remember this equation, final minus initial divided by initial times by 100. Let's first analyze the setup in the problem. It says that the following data was gained from an experiment in which this solution was placed in dialysis tubing bags and soaked in different solutions. So these are the solutions that they're soaked in. So the first solution is 100% pure water. So let's diagram it in this box that says setup. So we have a beaker, 0 molar solution, so it's 100% water, and the dialysis bag was placed inside and it contained a solution that was 0.4 molar. From this information alone, you should be able to predict which way water is going to move, and you should be able to explain why water is moving in that direction. So there's more solutes in the dialysis bag compared to the outside solution, so water would be moving in. And I've written an example explanation for you here. So this is a good example of an explanation for that change in mass. So now let's calculate the percent change in mass. The final mass of the bag was 4.5. The initial was 4 grams. So you can tell that water is moving into the bag, and then we divide by initial, 4 grams. So we have a change of 0.5 divided by 4. That gives us 0.125. We have to times by 100. That gives us 12.5. So this means that we had a 12.5% increase in the mass of the bag. Now this is a positive increase because the mass of the bag went up. So technically there's a plus there in front of that 12.5%. If we end up with a negative percent, then that simply means that the mass of the bag is decreasing because water is being pulled out of the bag. Now let me just reason through this calculation. If I want to know the percent change of the bag, then we're going to put the change on top. And it's the change of the original. And the original was 4 grams. So let's just think about what the change is. So just compare these two numbers. The change is 0.5, right? So we have a 0.5 gram change over 4 grams. If you divide and multiply by 100, it's going to give you the same answer. And since the change was a positive change, mass goes up, then just make sure that you put a plus in front of the number. So let's look at the next example. And we have a change, and it looks like it's dropping 0.4 grams. So our change is 0.4 grams, and it's decreasing, so let's put a negative in front of it. The original mass is 3.9 grams, so we times by 100, and that's going to give us our negative percent change in mass. So let's solve that just using this equation right here. Our final is 3.5 grams minus the initial, 3.9 grams, divided by our initial, which is 3.9 grams. And when you do the math for the top, that's going to give us a negative 0.4 grams divided by 3.9 grams. And that's a negative 0.1. Times that by 100, and then we have negative 10%. So that just simply means that the mass of the bag decreased 10%, and that negative is just an indication that it was a decrease, not an increase. Just diagramming what this looks like. So if I give you a scenario, sometimes it helps students to diagram it. So again, in this situation, we have a 0.6 solution on the outside. We're putting that 0.4 molar solution inside. You know that water should move out because there's a higher solute concentration in the outside solution. And then you should be able to explain that. So organisms are constantly trying to regulate their water and their solute intake, because if the concentration of water or solute reaches abnormal levels, then that can affect the movement of water in and out of cells and could lead to cell death. So organisms are always regulating water and solute concentrations. So you should be able to indicate what's going to happen to cells if they're put in different types of solution. And it's going to be different whether we're talking about animal cells or plant cells. So if we take an animal cell and we put it in a hypotonic solution, then that means the outside is hypotonic compared to the inside is hypertonic. Water moves to where there's more solutes or into the hypertonic solution. And for animal cells, since we're lacking that cell wall, then our cells can rupture. And whenever animal cells rupture or lyse, this is called cytolysis, but it's pronounced cytolysis. Cyto means cell, and lysis means to rupture. If we put our cells in a hypertonic solution, then the inside is hypo. Water is going to move out to the hypertonic solution, and our cells are going to shrivel up. And when they shrivel up, it's called crenation. And to help you remember this term, I like to think of cremation. And someday if you're cremated, you're going to get a lot smaller than what you are right now. So think of crenation is when cells shrink. Cremation is going to make us shrink someday. So processes in multicellular animals are always working to make sure that the outside solutions of our cells are isotonic compared to our cells, so that our cells aren't bringing in water or losing water, and they can maintain a normal shape. Now, protists have different ways to help them maintain homeostasis because they're just single-cell organisms. So if we have protists that are living in a hypotonic solution compared to the cell itself, then that probably means that they're a freshwater organism. Because in a freshwater environment, there's going to be more solutes inside the cell compared to the outside. And so water is going to be going into the cell. And we mentioned that protists that live in freshwater environments have these contractile vacuoles, which you see there, and they fill up with the excess water, and eventually they're going to constrict, and that's going to move water back out into the environment. So these cells are going to have contractile vacuoles to help them maintain homeostasis. Protists that live in saltwater environments, they have ion transporters to help them maintain homeostasis. So what that looks like for them, let's draw a protist and let's put them in salt water. And it's going to be hypertonic because there's going to be more solutes in it, or a higher solute concentration compared to the cell. And so the cells are always losing water to the environment. So what these cells do is they actually transport in ions. Let's label this first arrow with water so that you understand this second one is indicating ions or solutes being brought in. So they bring in solutes so that they can become more isotonic compared to the salt water, so that they're not always losing water to the environment. Now, plant cells are different from animal cells because our cells need an isotonic environment. The best condition for plant cells is to be in a hypotonic environment. So here's a situation where the outside is hypotonic compared to the cell, it's hypertonic. And when you put water on plants, so if you just water plants using tap water, then that's going to be hypotonic compared to the plant cells. So the water from the outside environment is going to move into the plant cells, and these cells are going to get really tight. They're going to develop this high turgor pressure. Now, turgor pressure is pressure due to water in the cell on the cell wall. So animal cells don't develop turgor pressure because we don't have a cell wall. So this hypotonic environment is going to create cells with high turgor pressure. And that means that these cells are going to be very tight. They're going to be very crisp. If you bite into a stalk of celery and those cells have high turgor pressure, it's going to be very crunchy because they're full of water. And the fact that they're tight allows these plants to keep their structures upright. So their leaves are going to be upright and their limbs are going to be upright and they're not going to fall down to the ground. Cells that are in a hypertonic environment, so here's the hypertonic environment, and we can tell that because this cell is losing water. So the inside was hypo. And whenever plant cells shrink up, whenever their cell membranes pull away from the cell wall, so here's a situation where we have the cell wall. It's still intact and it's still there, but the cell membrane pulls away from the cell wall as the plant cell loses water. And so these cells shrink up, and this is called plasmolysis. So let's review those three terms again. Whenever animal cells burst due to taking in too much water, that's called cytolysis or cytolysis. Whenever animal cells shrink up, remember cremation, but it's called crenation. Whenever plant cells shrink up, it's called plasmolysis. And there's no term for plant cells bursting, because remember, they will not burst because they have that cell wall. So eventually the cell membrane is going to get pushed out, but it's going to be pushed out right directly against that cell wall, and it can't continue to expand and rupture. So cells that have low turgor pressure are going to be very limp or very flaccid, and this plant is going to look wilted. Its leaves and its stems would have dropped towards the ground.